Aloha, this is Roger Brewer with the Hawaii Department of Health. This is a recording of a presentation I gave to the South Carolina PFAS Affinity Group on November 3rd, 2022. The title of the presentation is Field Study of Perfluoroalkyl Substances, PFAS, in Wastewater Treatment Plant Influent, Effluent, and Biosolids, Preliminary Data from Hawaii. It's very similar to a presentation I gave to USEPA a few months earlier. Just added in a few more notes. We're especially looking at leaching of PFAS from biosolids. Had a lot of talks with people in between. So in this study, we went to 3 wastewater treatment plants in Hawaii. We collected samples of influent, effluent, and the biosolids and tested them for PFAS. We're especially concerned with the use of effluent for crop irrigation and potential impacts of drinking water, aquifers, and also discharges to aquatic habitats. Now in Hawaii, not much of the effluent is actually used for irrigation, and it's not really impacting drinking water. We haven't found any major PFAS problems in drinking water so far in Hawaii, but still a useful research project. For biosolids, especially concerned about potential direct exposure risk where biosolids are applied to parks, and in some cases, agricultural fields here. Most of the biosolids are actually disposed of in landfill. And also concerned about potential leaching of PFAS from biosolids and impacts on groundwater. And another potential concern is uptake into plants in agricultural fields have been amended by PFAS. We first developed action levels for soil and groundwater in Hawaii for PFAS in 2020 after PFAS were found in the groundwater under a fire training area in Kahului, Maui. Wasn't impacting a source of drinking water, but there is concern the plume could be discharging into the adjacent beach area. I gave a similar presentation on this topic to the South Carolina group in 2020. And this presentation was a follow-up where we actually went out and started testing PFAS at wastewater treatment plants. So in 2020, in that presentation, I'll review some of that today. We coordinated first with several states and federal and European agencies trying to compile information that they'd done on PFAS, especially wanted to compile physiochemical constants and toxicity factors, which we're able to do, and I'll review in a minute, for 18 PFAS compounds. And then based on this, we were able to develop direct exposure action levels and leaching action levels for PFAS in soil. And for groundwater, we were able to develop toxicity-based action levels for drinking water, and also compiled action levels for aquatic habitat toxicity. So in this presentation, or in this study, we, as I mentioned before, wanted to go out and actually start collecting data, and we started at wastewater treatment plants in Hawaii. A few references are guidance we published on PFAS action levels, most recently updated in 2021, interim soil and water environmental action levels for perfluoroalkyl and polyfluoroalkyl substances. We'll probably update this again early next year based on the results of this study. We definitely need to add a few more compounds for, especially for precursor compounds that we identified in wastewater treatment plants. Our guidance document for development of the action levels is in the second reference here, evaluation of environmental hazards at sites with contaminated soil and groundwater. This just goes through the models and the assumptions that are used to develop the action levels. And then the third guidance, especially important because now we're getting out in the field and collecting samples. This discusses sample collection, processing, and testing. This is in sections 3, 4, and 5 of our office's technical guidance manual. And you can see the link there. And then the last reference, IPRC 2020, is a really useful overview of perfluoro and polyfluoroalkyl substances. Now, our previously recorded webinars and other information is posted to our PFAS webpage. You see here the link. And also we have a separate webpage where we post recordings of our webinars. I didn't spend a lot of time on the background of PFAS in this presentation. I think most people are familiar with it, but basically PFAS are pretty simple compounds. The names sound really complex, but it's actually fairly simple. Just a chain of carbon with fluorine attached to it, and then on the end of the molecule is some other family group, either sulfonic acids or sulfonates or carboxylic acids and carboxylates. Now, the second term I was saying here refers to the anion form of the compound. This is where the hydrogen atom essentially gets stripped off of the PFAS when it dissolves in water. That's something we're especially concerned about because this is the type of compound that you're most likely to find in the environment. When you do this, make this change to anion form, it's going to change the fate and transport mechanisms of the compound slightly. So we have to take that into account when we're developing action levels, especially for leaching concerns. So you see different types of PFAS here. You should be familiar with most of the ones up top. And we listed a few other types of PFAS down at the bottom, especially the bottom right. Keep this one in mind, this 5,3-FTCA, or these fluorotelomere carboxylate compounds, because they ended up showing up quite a bit in our wastewater treatment plants. This is a list of the 18 PFAS where we were able to compile toxicity factors and physiochemical constants for. Again, this is discussed in more detail in a previous presentation in 2020, 2021. So you can see the different names of the compounds here. They're all in the anionic form. And again, because this is assumed to be most prevalent in the environment. And the abbreviations at the end, you see a negative sign at the end of the abbreviation. Again, this just signifies that we're discussing the anion form of the compound. And the references for the sources of the toxicity factors and physiochemical constants, they're included in the 2021 memorandum I discussed at the beginning. It's a mixture of state, federal, and again, European agency guidance is published. So based on this, we were able to develop action levels for PFAS in groundwater. So 2 sets of action levels. One was based on drinking water toxicity. You see the numbers here, should look fairly familiar. They're in the nanogram per liter range. And in the second column are chronic aquatic toxicity action levels for PFAS. These are based on a really useful compilation of data and studies put together by the Washington Department of Ecology in 2021. Now you can see some blanks there for a lot of the PFAS. When we don't have an aquatic toxicity action level for a PFAS compound or for any compound in our guidance, we default to the drinking water toxicity action level just so we have a number there. In the reality, if you think you have a potential concern with aquatic toxicity, we haven't done this here in Hawaii, but we plan to do some field studies. The best thing to do is get a sample of the groundwater and then do some type of bioassay study on it to see what the effects actually are on a more site-specific basis. That's the groundwater action levels. And again, drinking water toxicity-based action levels much lower than chronic aquatic toxicity action levels. These are our soil action levels. So we just have 2. One is for direct exposure. This is based on non-cancer hazard to young children. Assume a body weight of 15, relative source contribution of 0.2, which just means we're assuming they're getting PFAS, 80% of the PFAS they're exposed to are from other sources, and a hazard quotient of 0.5. This is just done to take into account potential cumulative health effects. So you see the numbers here all in the low ppb range, single digit to double digit ppb range for the most part. In the second column is the action levels for leaching of PFAS out of soil and the impacts to underlying drinking water aquifers. This is based on a sea soil model. It's a generic model we use in our guidance. It assumes groundwater is very shallow within 10 or 20 or 30 feet of the source. Such we know it's too conservative, but it's a good start just for a screening level. You can see these action levels are order of magnitude lower than the ones for direct exposure. This is what we've been hearing from other states. Don't see that necessarily big of a problem with direct exposure to PFAS in soil at a lot of sites, especially where biosolids have been applied, but they all get flagged for potential leaching concerns when you run these really simplistic models. That's one thing we wanted to look at is how accurate are these models. And my gut feeling has always been that they're way too conservative. But there's ways we can look at that site-specific basis. So additional PFAS that we detected in the wastewater treatment plants that we haven't been able to compile toxicity factors and physiochemical constants for. You see them listed here, different sulfonates and such. So really the last 2 are especially an issue that we're looking at because again, you'll see in a minute, we see a lot of these fluorotelomere carboxylates in the biosolids and even in the influent and the effluent at wastewater treatment plants. This is something that it could be overlooked if you're only testing for a typical short list, especially if you're only looking for PFOS and PFOA. You could be missing most of what's actually there. So let's go look at the wastewater treatment plants. We at this point have only collected samples from 3 wastewater treatment plants in Hilo on the Big Island, Lihue on Kauai, and in Kihei on Maui. These are all fairly small wastewater treatment plants, just 1 or 2 or 3 few million gallons of influent a day. And then they're generating, you can see here, 30 to maybe 150 tons of biosolids a week. Not that much. If you're not used to thinking in terms of volume, the dump trucks you see going down the road with dirt or something in the back, I'm usually 10 or 20 tons. So we're talking about 1 or 2 or just a few trucks of biosolids a week. Again, most of the biosolids at these wastewater treatment plants, in fact, at all 3 of these wastewater treatment plants, all the biosolids are going to a landfill. One point is for the treatment plants themselves, landfill leachate's not being disposed of at any of these wastewater treatment plants, and we know that for a definite fact. These treatment plants vary some. I'm not familiar at all with how wastewater treatment plants work, but I added a few notes here on the side. They're all getting influent from residential commercial areas. Really no manufacturing going on. Not much at all in Hawaii to begin with, but especially in these areas. And you can see the different types of processors they have at the treatment plants. The Hilo plant is really old, and it's pretty dilapidated, and they're trying to get it replaced. It's something to keep in mind when you're thinking about how PFAS alter the relative makeup, alters between the influent and the effluent and the biosolids. I think that's going to show up in our data. One thing with the biosolids is this stuff is really like a sponge. That's why I have the sponges up there. The biosolids are typically 80 to 85% water, and that's something that comes up in terms of leaching that we need to think more about. We have plans to collect samples from 5 other wastewater treatment plants, all on the island of Oahu, where Honolulu is, sometime hopefully in the next few months. Before we ever go out in the field to collect samples, one thing we really emphasize in our guidance and our studies here is first you write down the specific questions that you want to address in this study. So it's a very methodical approach. So the 2 investigation questions we established for this study, first is what is the average makeup and concentration of PFAS in the influent, effluent, and biosolids during a 24-hour period at the wastewater treatment plant. So it's really important that we establish a time for it. And this is what we refer to as the decision unit. So we have a specific volume of influent and effluent and of biosolids generated during a 24-hour period that we want to get a sample of. And our objective, we're not concerned about variability during those 24 hours or during an hour or during 30 seconds or whatever. We only want the concentration for the effluent and influent and biosolids for that specific volume. So we just need one number, which means we need one good sample that's representative of that time period. So the data need, that's all what the risk assessors might ask. The data need is what you tell the field people. I need the mean concentration of PFAS in the volume of influent, effluent, and biosolids generated during a 24-hour period. And the second type of data we need, I need the relative amount of PFAS sorbed to biosolids versus leached into solution in some type of leaching study. So we have our questions, we have the data needs. The next step is, okay, how do we collect representative samples to address this? So the water is actually pretty simple. All the wastewater treatment plants are set up with these 24-hour samplers. So they'll collect a single sample of all those millions of gallons of influent and effluent flowing through the wastewater treatment plant. So as the influent is coming through in a pipe, then a sample is being slowly collected over that whole 24-hour period. So you end up with a liter or a few liters of water that's representative of millions of liters that went through. So that's the way to collect a sample. It's a good sample. With the biosolids, we want to do the same. It's the same idea, same approach. So at each of these plants during a day, let's say they generate 10 cubic yards of biosolids. So you can see in this container in front, the bottom center of the slide. So we want to get a representative sample of that, just one sample. We want one number. What's the mean concentration or the true concentration of PFAS in those biosolids? Ideally, we'd take the entire mass of biosolids, all 10 tons, send it to a lab, they extract the entire mass and give us a number. What's the concentration of PFAS in there for each compound? We can't do that, obviously. It's not very practical. So we have to collect a representative sample. Well, how do we do that? If this is addressed in our guidance, you're going to collect a single multi-increment sample from about at least 30 points within those biosolids. So we lay a grid across the biosolids and we'd collect, say, in this case, maybe even 50 grams subsamples from each one of those points and combine it all together into a single sample. That final sample should weigh 2 to 4 kilograms. Our default guidance is 1 to 2 kilograms. In our first study, that's what we did, but then we realized this stuff's mostly water, and after they air dried it, there was not much left. So in our next studies, next plants we're testing, we're getting 2 to 4 kilograms. So instead of 5 to 10 gram subsamples, I note here, never mind, that's at the laboratory. So we get big samples, and to move on to that, so we get a representative sample in the field, we send that to the laboratory. Well, they can't just open up the bag or the jar or whatever and take out a pinch and test it. They need to get a representative subsample. And the only way you can do this, first they're going to air dry the biosolids. And our guidance says also to sieve it to get the less than 2 millimeter particles. It's not going to be a problem for this stuff, but you might get some clumps of clay or hard material you're going to have to mechanically break up. You do this, air dry it, break it up into small particles, and you're going to collect a single 5 to 10 gram subsample from this spread out dried biosolids in the same way you did it in the field, from about 30 points within that with a small, very small spoon or something. So now the subsample you collect from the sample at the laboratory is going to be representative of the sample, and the sample you collected in the field is going to be representative of that targeted mass or volume of biosolids. So that's the field methods. And the laboratory we use was SGS, Access Laboratories, Analytical Services. I believe most of the tests were done in their Florida lab. Those are the objectives. We spent a lot of time here thinking about what's the question, what do you want, and how you're going to get the data before we actually go out in the field. If you're not familiar with decision unit and multi-increment sampling methods, the first reference here is discussed in a lot of detail in our technical guidance manual. I think we're one of the few states that has a field team, so we actually go out and do a lot of our own sampling. So there's a lot of our own field experience, but especially compiled from consultants and other people who have experience in doing this type of sampling in the field. It's included in our guidance manual. And we have several webinars and training courses and such posted to our DUMIS webpage. And then the last document here, it should be coming out soon. Hopefully it's end of this year. I've been working with the Chinese Academy of Sciences for the last 5 or 6 years and doing work over there. They have a lot of experience in contamination, as you might imagine, but really excellent in the field. So myself and Dr. Sung Jing of the Academy of Sciences, we took Hawaii's guidance, translated into Chinese, rearranged it to make it easier to use, and then expanded it quite a bit to turn it into more of a textbook and to address these mega industrial sites or all the way down to a single gas station. But this document, this guidance should be published by the Academy of Sciences sometime this year. And as soon as they put a final number on it, it's pretty much ready to go. Then I'll take the English version we translated it back into and I'll post it to our webpage and give a presentation on it. That's how to collect samples. The leaching test, again, we're especially interested in. So there's 2 ways to test for the leachability of PFAS or of any compound contaminant, especially 2 ways from soil. The first is the synthetic precipitation leaching procedure, SPLP. It's very similar to TCLP. If you're familiar with that, you take basically a spoonful of the material, a representative spoonful, of course, and you put it in 2 liters of fake rainwater and you shake it up and agitate it for 2 days or for 18 hours or something. And then you test the water, and then you can estimate the mass of PFAS that was stripped from the biosolid sample that you put in there. And you've already tested the biosolid sample itself, so you know the original mass. So from this data, then you can compare the ratio of the mass of PFAS that stayed sorbed to the soil versus what went into the water. This is referred to as the sorption coefficient, or KD value. We'll look at that again in more detail. That's what these SPLP tests are intended for. Now, the problem we have, and I definitely used to do the same thing, is you get the SPLP data from the laboratory. It gives you a concentration in the solution. You actually can't compare this to your groundwater action level. You can't directly compare it to it. That's not what the test is intended to mimic. You have to calculate a sorption coefficient. Then you plug the sorption coefficient into an equilibrium partitioning model, basically a leaching model. And it's discussed in our guidance on batch test leaching. So we'll look at that. We ended up doing a lot of SPLP tests on our biosolids. The second ideal way, which we're still working on, is the soil column leaching test. This is what we really want. We're trying to mimic what goes on in the field. Let's say you have an agricultural field, you've amended it with biosolids, and you want to know what's going to leach out of this field and get into groundwater. Whether step one is to get a representative sample of the soil, amended soil, the biosolids from the field, and you put it in basically a PVC tube or some type of tube that you see in the picture here. And then you pump water up through the bottom of the tube and it comes out the top. You do it from the bottom up so you can push out the air. And then there's your leachate coming out the top of the tube. So it's a well-established test. They use this a lot in agricultural work to look at pesticides, but it's fairly new to the environmental world. And one thing we have to think about here is first, the original leaf method, 1314, the tube is too small to get a 2 to 4 kilogram sample in. So you can't get a representative sample in the tube under the default methods. We have to modify that, make a bigger tube. Not that big a deal. The consultant who did this for us with the SGS lab in Ontario, Canada, he knows about this in detail. He just made a bigger tube. So that's the second part. We got the sample in the tube. The next part, well, how much water do you pump through? Well, here we want to simulate infiltrating irrigation water over the field. We know how much water is used to irrigate fields. On average in Hawaii, it's 120 centimeters per year. So if we want to mimic 1 year of irrigation in this setup here, then we would pump 120 centimeters of water through this tube and collect the sample, collect the leachate at the end, and there's our 1 year's worth of leachate. We test it and get one number. That's the way to do this. The current leaf method recommends that you test the leachate 9 times. You're developing a curve for the mass coming out over time. We don't really need that. It's not really the question that we're asking. We want to know, say, over a 1 year time period, what's the concentration of PFAS in that volume of leachate. We could also look at first flush. What's the mean concentration of PFAS in leachate generated from the field over a 1 month time period? So in that case, we're looking at maybe 10 centimeters of water pumped through the soil. So that's the way we're setting up the test. We didn't actually run any tests. We ran some practice tests on some PFAS. You can see on the right-hand photo up there, the leachate came out really murky because we put in pure biosolids, which that was a mistake. So it would have to be filtered. So we're going to fix the test, modify it, and then run it on some of the samples we collect next year at these other wastewater treatment plants. So in the end, we'll collect 2 samples. I think we'll do a first flush sample, the first few centimeters of water that goes through the column. And I bet that's where we see the highest concentrations of PFAS and mass getting stripped out. And then we'll keep running it, say, to simulate 1 or 2 years worth of infiltrating irrigation water, and then collect that as a sample and test it. So look at more long-term impacts. That was the leaching, and so in the end, we're kind of stuck doing the SPLP batch test, calculating sorption coefficients. That's still very useful. It gives you kind of a quantitative, relative idea of how sticky this stuff is, how well it sorbs to the soil. And this is discussed in our batch test guidance. You see here at the bottom how to run the batch test. And again, the sorption coefficient, the KD factor, it's just the ratio of the concentration of the contaminants that stays sorbed to the soil divided by the concentration of the contaminant that went into solution. And when this ratio starts to hit 20, then that's an indication that the contaminant is really tightly bound to the soil and doesn't really pose that big of a leaching hazard. So you can see things like MTBE, PCE, atrazines, a pesticide, they don't stick to the soil at all. They go straight through. They've got a KD factor of less than 1. And that's why we see a lot of plumes generated from these contaminants. They don't stick to the soil or other particulate matter. You get to something like diesel, which is more moderately sorptive. It can still leach from the soil, but it likes to stick to organic material in the soil. So a little bit less mobile comes to leaching. Then you get compounds like dieldrin, where the KD factor is getting up between 10 and 20. Well, dieldrin is not that leachable from the soil. You'll still find it in groundwater, but you don't typically generate very large plumes because it has a very difficult time migrating. The lab chemist, he said one way to think about this is think of each molecule of this stuff like a ping pong ball that's covered in Velcro. The higher the KD factor, the more Velcro that's stuck to it. By the time you get up to something like DDT, they're like Velcro, completely Velcro covered ping pong balls, and try to shove that through the soil thinking of all the organic material. Organic carbon is the other side of Velcro. It's not going to get very far. It might break away from the soil and go into solution, but real quickly it's going to reattach. That's why you don't get big plumes of DDT and PCBs, especially when you get to benzoate, and dioxins and such. So that was just some background information before we start looking at the data on leaching and such. So let's jump into the data now. So first we'll look at the HIDOH wastewater treatment plant. All these charts you're going to see are the same. The bottom left-hand corner is a pie chart for the influent. We're just looking at the relative makeup here. And then upper left-hand is the makeup of the effluent, PFAS. Upper right is the makeup of the biosolids. And on the bottom right, this is what went into solution in the SPLP batch test solution. So let's start with the influent here at the HIDOH wastewater treatment plant. Well, here was the first surprise, mainly because we hadn't done this before, but dominated by this compound FTCA, these fluorotelomere carboxylates, 41%, the 5,3. And the next most dominant compound was the hexanoate, perfluorohexanoate, 21%. So that really makes up most of it, most of the influent, the PFAS and the influent. Well, we don't have any toxicity factors, physiochemical constants for FTCA. So this immediately threw up a flag to think about. Look at the effluent. It looks pretty much like the influent. And this is at the HIDOH plant. It's really old. It needs refurbishing. So it doesn't look like much is happening to the influent as it goes into the plant and comes out. The effluent, the concentrations are even close to identical. So keep that in mind when we look at these other sites. Then the biosolids, there's that FTCA again, dominating the biosolids, the 5,3, but there's still a lot of the 7,3 FTCA. And then this other compound, the NMEFOSE. So think about this when you're out doing testing at wastewater treatment plants. And I know in a lot of states still you only test for PFOA, PFOS. You're missing most, if not all, of the stuff, the PFAS are in the biosolids, and pretty much a large portion of what's in the influent and effluent, these plants. So that throws up a flag that we may be missing and underestimating risk associated with these. Let's look at the SPLP solution, the bottom right. You see all the red there. Well, first, the solution itself dominated by, here's this FTCA again, 5,3 FTCA, more than 3/4 of the PFAS in the solution. You see it though, they're all in red. That means that the KD factor, that sorption coefficient that we calculated for these compounds, is all over 20, which means it went into solution, but this stuff is still really sorptive. It probably wouldn't get very far in the leachate itself before it resorbed to other organic material, at least in the biosolids. So that's interesting. It'll strip out into solution, but it's not going anywhere, according to this, just looking at KD factors. I'm not sure how much to trust these. It's something we want to think about more. Here's the LeHue plant. So the influent looks pretty similar. There's the FTCA again and the hexanoate with the PFOA, a little bit of PFOS, such in the influent. Similar concentrations. Effluent though, look at the effluent here. And LeHue and HIDOH, very similar population size, very similar commercial, industrial, commercial residential areas, no industrial really going on. Again, there's no landfill leachate going into these treatment plants. But in the effluent at LeHue, all the FTCA is gone, or at least it's not detectable. You see the detection method reporting level there, 10 nanograms per liter. Maybe there's a little bit there, but all of a sudden we get the pentanoate, this PFPEA, 32% dominating the effluent. So what's going on there? It must be something going on in the process of the wastewater treatment plant. It's converting some of these compounds or stripping them out. I don't know. Something we need to think about. Now the biosolids, so they look pretty similar to HIDOH. There's the FTCA again. You get some PFOS, but still quite a bit of the 7.3 FTCA with the 5.3 FTCA, a little bit higher concentration, but still talking about micrograms per kilogram. So that looks similar. And you see the other, which we also saw in the HILO plant, this long list of other stuff that shows up in the biosolids, where this turns out this is all the really sorptive PFAS compounds, and they're all stuck to organic material. And just to flip back, anyway, there's LEHUE. So that if we looked at HILO plant, you see the same thing. SPLP solution again, what's getting stripped out in this leaching test, where there's the FTCA again dominating what's in leachate. This time you see it's in black, the number there, 67%. That means the KD factor was less than 20, so it's more mobile. In this case, it was 19, so it's still pretty high absorption coefficient. It's not that mobile as far as being stripped out of the biosolids based on this. And then you see the other compounds, the other FTCA and these other compounds get stripped out. But again, for the most part, it's red. The KD factor is over 20, not that mobile in reality, except for the hexanoate. Keep an eye on this because this actually shows up a lot in groundwater. In this case, the KD factor for the hexanoate was well below 20, which suggests it can be fairly mobile. And some of the other compounds you see at the bottom in black, the perfluorobutanoate and the perfluoroheptanoate, both in black down there. This means the KD factor less than 20, they're going to be a little bit more of a concern for leaching. Here's Kihei plant, and there's the hexanoate again. The influent doesn't look that different from the Hue plant. This is Kihei on Maui. Again, dominated by the hexanoate, the butanoate and such. There's some PFOA there, but there's also a lot of the pentanoate, the PEA. But then you look at the effluent again, and the effluent looks kind of similar. There's all that pentanoate again in the effluent and the hexanoate. This plant had activated sludge. It was one of the more recently constructed wastewater treatment plants, refurbished. It's a little bit different there. And there's a higher concentration of PFAS in the effluent than the influent. I don't know if this is just due to variability. It could be. We're talking about nanograms per liter. It's not going to be the exact same every day. But I'm not sure if they collected the influent and effluent samples at the same time here. They probably did. But either way, it looks like there could be more PFAS in the effluent than influent, which is something that some people during the presentation with South Carolina folks pointed out. Could be a process of changes going on within the wastewater treatment plant as this stuff's breaking down. Biosolids there it is again, that 5.3 FTCA, 7.3 FTCA. So, there's some other stuff in there, but these compounds still dominate. And then leachability, look at the bottom right, SPLP solution, same thing. There's that 5.3 FTCA. That's what's getting stripped out, but again, it's in red, which means the KD factor is over 20. It may come off for a few seconds or something, but in theory, it's going to absorb again pretty quickly. But there's the hexanoate again, you see in black in the leachate solution, which means absorption coefficient is less than 20, so that is a potential leaching concern. So those were the three plants that we looked at, Hilo, LEHUE, and Kihei. Now just comparing the data for the influent, especially the effluent data, the water data to our action levels, this was interesting. None of the PFAS exceeded our action level for drinking water toxicity, and none of the PFAS in the effluent exceeded the action level for aquatic toxicity. So not that big of a concern in terms of the effluent coming out of these wastewater treatment plants. For direct exposure, this is the PFAS and the biosolids. At Hilo, none of the PFAS compounds exceeded our direct exposure action levels. But at LEHUE and Kihei, then the PFDA, the decanoate, and the dodecanoate slightly exceeded the action levels, but not that much. If you did a risk assessment, your cumulative hazard index is going to be less than 1. So probably not that big of a risk from direct exposure. But what gets flagged is the leaching action level. So the compounds that we have action levels for, the decanoate, you see all the others here, PFOA, PFOS, depending on the plant, they all exceeded the action levels in the biosolids for potential leaching concerns. We also looked at the precursor compounds, the FTCAs, and these other compounds. In this case, we just made an assumption just for illustration purposes. We compared those data for those compounds to the compound that they would eventually break down to, like the 5,3 FTCA, if I have it right, would break down to the pentanoates. And they exceeded the action level for leaching concerns. So we have some leaching concerns for our main list of compounds, but also these precursors, again, getting flagged for potential leaching concerns just by comparison to the action levels for leaching. And again, in this case, we're comparing it to action levels addressed to protect groundwater as a source of drinking water. So once these get flagged for potential leaching concerns, that means take the sample, run an SPLP batch test on it, and look at the absorption coefficient again to see if it's above 20 or how mobile this stuff really could be. So we talked about that. This is kind of a busy chart, but this HIDOH wastewater treatment plant, this is a comparison of the predicted absorption coefficient versus what we measured in the field, the KD factors. We can predict the absorption coefficient by taking the absorption coefficient for that individual compound, and you multiply it by the percent organic carbon in the soil, which in this case for biosolids, we assume 60% organic carbon in the biosolids. We didn't get data, but that's typically in the ballpark. So I wanted to do in these figures, just get an idea, is the predicted mobility, leachability of these compounds, is that what we're seeing in the field also? And it turns out it does. Actually, it works out pretty well as far as putting it in relative sense. So the further up and to the right you go, the more absorbed the compounds are to the biosolids. You see in the upper right-hand corner, the PFTDA, that tetradecanoate, really high absorption coefficient, predicted and measured, 10,000 or so, which means this stuff isn't going anywhere. It's not going to get leached from the soil or from the biosolids. Then as you go down to the bottom, toward the origin, then the KD factor decreases, predicted and measured. And you can see the compounds that were predicted to be more mobile with lower KD factors, pretty much what we measured in the field, things like the butasulfonate, PFBS. And then there's the pentanoate, the hexanoate, butanoate, all these compounds you see toward the bottom. So we would predict, just based on their absorption coefficients, these things would have a low absorption coefficient overall, or KD factor, and be fairly mobile. That's what we saw in the field. But again, in this case for HIDOH, KD factors are still over 20. So they don't seem to be very mobile, including for FTCA. That was HIDOH. Again, the main point here is the predictive mobility is pretty much what we saw in the field, in the right ballpark. You still want to get site-specific data, but leaching models actually do seem to work to start with. The HUI, similar thing, predicted versus measured KD. So here you see some of the compounds that are predicted to be more mobile, lower KD factor. Actually, they were in the field. This PFBA, butanoate, the heptanoate, hexanoate, even the butasulfonate, PFBS, predicted to have fairly low absorption coefficients, and sure enough, that's what we measured in the field. In this case, the absorption coefficients were, they're pretty close to what's predicted, but still lower. And in some cases, they're actually lower than what would be predicted, like the PFBA and the heptanoate. So the measured absorption coefficient, in some cases, much lower, which means it's more leachable than we might initially predict. A lot of variability between these plants, different wastewater plants. Here's the last one, Kihei, same thing. You see in the upper, as you go further up and to the right, you get the longer chain PFAS compounds. They're more absorptive. They're predicted to be more absorptive, and we measure them in the field to be more absorptive. So it's all working there. As you get down toward the origin, the lower left, you see these same compounds, pentanoate, hexanoate, butasulfonate to some extent, and butanoates, heptanoates, and such, predicted to be more mobile, have lower KD factors, and that's what we actually measured in our test. So science actually works sometimes. That's all kind of complicated. I tried to simplify it in this compound. What we're thinking about, what are the PFAS most likely to leach to groundwater and migrate away from the source area? Well, this pretty much sums it up. See, from left to right, the hexanoate, PFHXA, we saw, there's a range of KD factor absorption coefficients depending on the wastewater treatment plant, but they're in that moderate to slightly mobile phase, or mobile range for the KD factors. And then you see the heptanoate, the butanoate, octanoate. As you get longer chain, they get more sorptive and your KD factors go up. And FTCA and such, again, we saw that a lot in the leachate samples. That's something I want to look at in more detail, the SPLP data. It's really coming off the biosolids initially, but it has a high KD factor, which suggests it's not going to get very far once it does. And you get things like the hexasulfonate in the upper right-hand corner that we just wouldn't predict to get very far at all as far as leaching out of soil and getting into the groundwater, either predicted or based on our field data here. This actually works out. Compared in 2020, we made a prediction of how these plumes might segregate over time. Let's say you dropped all these compounds, mixture of PFAS, say at a fire training area, whatever, in some source area, and the groundwater is flowing through it, picking up the PFAS and migrating away with a less sorptive, more soluble compounds are going to take off with the groundwater. They're going to move out to the leading edge of the plume. And these are listed in order of sorptive capacity. So we'd expect to see at the leading edge of these PFAS plumes the hexanoate. That's predicted and what we measured in the field with sorption coefficients. And then you see the pentanoate, and the heptanoate, and the butosulfonates, and the butanoates. They're all going to be out there in the leading edge of the plume. And back in toward the source area, you're going to get the more sorptive compounds like the PFOS, the PFOA, and the hexasulfonates and such. That's because they're just more sorptive to organic material in the soil. So I think we're seeing a lot of this on some field sites in Hawaii, and especially on the mainland we talked to. You see a lot of hexanoate in the groundwater, and that's just because it's less sorptive and moving away from the source area. Once you get back toward the source area, you're going to start seeing more of the PFOS, PFOA. So that's an issue, because if you're only looking at PFOS and PFOA, if you're in the leading edge of the plume, or even not that far from the source area, in some cases, it could just be a few hundred feet, you're not going to see any PFOS, PFOA, because they're too sorptive. They're like ping pong balls covered in Velcro trying to get through this aquifer. It's not going to go very fast. So you really have to look at these other compounds when you're assessing risk, investigating an area. That was it, the quick review of the data we had. There's still something going on with leaching. And so I gave a presentation to the USEPA group a month or 2 or 3 ago. And I was talking about how this stuff is really sorbed to biosolids. It's not that leachable based on the tests that we've been looking at. And then a professor, I think at the University in Oregon, she gave a presentation on actual field data where she collected samples of PFAS in the field, and especially of groundwater. And guess what? The PFAS are in the groundwater. So it's kind of odd. I was saying, well, they're not going to get in the groundwater, not very leachable. And she's saying, well, we're finding them in the groundwater. So how's that happening? I think one thing that might be going on, just to throw out an idea, is when these biosolids, remember, they're 80 to 85% effluent water, essentially mixed in with 15, 20% organic material. So it's like a sponge holding all this effluent water, spreading this out in the fields. One thing that could be happening is you irrigate the field or it rains on the field, then you're just getting diffusion from the PFAS that are already dissolved in the water and the biosolids into the infiltrating water, infiltrating irrigation or rainwater. And that's getting carried down through the vadose zone and getting into the groundwater. That could be one issue. This might be why I would predict anyway, and not just me, a lot of people I talk to, is when you apply biosolids to a field, you're going to get a first flush that's going to be your highest concentration within the first part of the irrigation, maybe the first month or something. And maybe a lot of the biosolids are coming out, but they're just getting stripped from the water that was already bound up in these biosolids, the PFAS were. That could be what's getting to the groundwater initially. But then over time, as you start, you've pretty much stripped out the PFAS and the water contained in the biosolids, and what's left is sorbed to the biosolids pretty firmly. So you're going to get a pretty quick drop off, I would predict anyway, in the mass of PFAS that's going into the groundwater. Something to think about, and we're still working on this. We'll do more tests next year. We have to do some actual plant uptake and field studies, but I know there's a big issue. What do we do with all this sludge and these biosolids from wastewater treatment plants? Well, maybe the main concern for leaching is what's in the water, so you dry this stuff out. and it's going to get a lot more tightly bound to the biosolids. And then when you put it in the field, it's not going to leach out. It's much significantly reduce leaching concerns. I think we're going to try this out in some leaching experiments next year, where we'll put in undried biosolids from wastewater treatment plants and probably a soil column test and look at first flush and long-term impacts in the leachate. And then we'll dry the biosolids, mix that in with the soil, and then run a leaching test on it. And I bet that the first flush from the dried biosolids is going to be way, way lower than the first flush we see from the wet biosolids. Who knows, might be completely wrong, but just something to think about. So that was a presentation. We had lots of great questions at the end. I tried to address a lot of the questions and comments that came up during this presentation, especially for future work. Again, the study we did, single day, just a day in the life of a wastewater treatment plant. We go back the next day. In reality, it's probably going to be fairly similar. There might be some variability. I think especially where you're in an area where you have manufacturing facilities or other things going on, it could change during periods of heavy rainfall, light rainfall, and such. We need to come up with toxicity and physiochemical factors for precursor compounds, because that's what's probably driving risk in all aspects of these wastewater treatment plants, not just the PFOS and PFOA, even these other 18 compounds we have action levels for. Something to think about. I assume we're going to look at what the final breakdown compounds could be. And the influent effluent, the makeup is variable between wastewater treatment plants to some degree. See a lot of 5,3 FTCA in the influent at 2 of the plants, not at all related to landfill leachate. We're 100% sure of that. So where is it coming from? I'm not sure. There were people in the call that knew a lot more about other sources of PFAS than I did, but I want to chat with them and think about this more. Now, the PFAS didn't exceed drinking water action levels in the influent or the effluent of the treatment plants here. Well, that's good news. So we're not really that concerned, I think, about treated wastewater. At some of the plants, we have a few that do R1 treatment and then irrigate with it. It shouldn't be a problem for impacts to drinking water because it's already below action levels for drinking water before it gets into the plant or as it's going into the plant. It's only leaching out of biosolids. Maybe that's a different story we have to look at. Same thing for aquatic toxicity. Concentrations in influent, effluent, PFAS way, way below aquatic toxicity action levels. We haven't looked at uptake in aquatic organisms. That's always a separate issue. Then makeup of the biosolids, it's actually real similar between the plants. That was interesting to see. I think you're just seeing all the really sticky stuff. The sticky PFAS are getting stuck to organic material in the biosolids, so maybe it's not that big of a surprise after all. The ones that are more sorptive. Most of the PFAS appear to be tightly bound to organic material, not significantly leachable, the KD factor above 20. But some of the shorter chain PFAS, they could be moderately leachable. Someone pointed out, who knows a lot more about compost than I do, was the compost, these biosolids break down over time, then the PFAS could be released from the compost. So you do actually get more enhanced leachability. That sounds like another really good field study. Maybe we'll work on that next year. So the contamination of groundwater from biosolids emitted fields due to PFAS, I still think it's in the first flush, this could be an issue with the water content of the biosolids. That's what's really contributing to it. Maybe if we can get rid of the water before you put the biosolids in the field, that would be something useful. And again, we need to look more at the effects of biological breakdown of biosolids and compost over time, which we have some field studies going on already planned for next year, including composting of compostable food containers, which is a big issue here. So that was the presentation. It was a really fun group to chat with, lots of new ideas, lots of work still to be done. Luckily in Hawaii, PFAS aren't that big of a concern. Here we have some fire training areas, but they generally tend to be located right against the coast. It's mostly a concern, discharges to nearby shorelines and aquatic habitats. There, the concentrations of several PFAS are well above our action levels for at least chronic aquatic toxicity. So we have to do something with those sites. We're probably going to go collect groundwater samples next year at some of the sites and do some bioassay tests and see if that really is a concern. I've never done that before, but I know several states, California, and I think Oregon and Washington have some guidance out on how to do that. So we'll do that next year. And also one thing I really want to start doing is looking at uptake of PFAS and produce grown in biosolids emitted soil. And I know some people are doing these studies, but when you do these studies, you have to be really careful how you collect the samples to get back to that. So in the field, you want to designate some plot area, and that's your decision. You want to collect one really good multi-increment sample from that area. And you'll get a 2, 3, 4 kilogram sample from, say 50 points or something. So then your sample represents the dirt in that area. When you grow your crop there, you want to get one really good sample of the crop. So you may want to test different parts of it. Maybe you want to test the roots, if you're planting carrots or whatever. Test the roots, the leaves, and the fruits separately, but you get one sample of each. And to be certain, that's the way you get representative samples. It's the way we should be doing the research, not with discrete samples. They're can be totally unreliable. That's why our data are always highly variable when we used to do that. Well, how do you know those samples are accurate or representative? You go back and do it 2 more times. Collect 2 more samples of the soil from totally different independent areas, 2 more multi-increment samples. So you have triplicate samples. You can calculate an RSD, relative standard deviation, and look at the precision of your overall sampling method. We have discussion of that in our guidance. Same thing for the produce. Collect triplicate independent samples of the produce and then compare the data for each sample. And if you're within an RSD of plus or minus RSD of 35%, then you can consider your data in general fairly representative. So there's some more topics to think about. And there's, again, a great group, great presentation. And my email is at the beginning slide, so we're always up for brainstorming and ideas. Thanks a lot.